Hello everybody, I hope you can hear me okay. My name is EMBL-EBI and I have been a scientific database curator for the past 6 years working at UniProt. And today I shall be guiding you through the UniProt database. So this webinar is largely aimed at new UniProt users or people who have very little experience using UniProt. So more experienced users may benefit from some of the upcoming webinars that we have. I do encourage you to ask any questions as we go along. We have Rosanna Zaru, who's also a curator, poised and ready to answer your questions in the chat box if you have anything to ask as I go along. Otherwise, do ask questions later on at the end of the webinar. And if you have any more complex questions related to specific areas of work that you're working on, please do email us at help@uniprot.org. So this is a basic guide of what I'm hoping to cover in this webinar. So really, I just want you to kind of get used to the concept of once you finish working on something and you publish your work, that's not the end of the line for your data. Your data can be accessed by other users, and other researchers can use bits of your sequences and your analyses to supplement their own work. So I'm just going to tell you how UniProt uses data that we get from sequence repositories, and also publications. So in this webinar, I'll show you how to search for proteins and how to get the most from a basic search. I'll show you how to access functional data, including information on where the protein's located and structural information if it's available. I'll show you how to access protein sequences, including any isoform sequences and sequence features such as amino acid modifications. I'll very briefly tell you about proteomes and how to access them. So a proteome is defined as the total number of proteins expressed by an organism. So I'll show you where you can find that information on our website, and I'll very briefly show you where you can find any mutations and disease annotations as well. So to begin with, what's UniProt? So UniProt is produced by a consortium that stems from a collaboration that still exists today between bioinformatics databases at institutes based in the UK, Switzerland, and the United States. And together we aim to provide a comprehensive, high quality and freely accessible resource of protein sequence and functional information. So that means we provide information on protein sequences, including isoform sequences and disease variants. We provide information on protein function, including subcellular location, any information on whether the protein's involved in any pathways, reactions, or interactions. And if available, we'll give you information on the structure, including access to any 3D structures. We also provide a range of analysis tools such as BLAST and multiple alignment. So where does the data come from? The majority of our protein sequence data comes from the International Nucleotide Sequence Database Consortium. And so providing accurate protein sequence data is our primary objective. So we're always updating this and we're always getting an influx of new sequences. But sequence data may also be imported from databases such as Ensemble and PDB, and researchers can directly submit protein sequences to us as well. And so once we get this sequence data, we analyze it, we organize it, and the way in which we organize it is into resources. So these are the main resources that we provide. I'll mainly be talking to you about the UniProtKB resource, which is the UniProt knowledge-based resource, and I will touch on the proteome section as I've mentioned. So the UniProtKB section is the central hub, and that contains, as well as sequences, it contains functional and structural information. The proteomes portal provides we provide proteomes of completely sequenced genomes. So I'll show you later how you can access those. UniProt is a non-redundant database that's an archive of most of the publicly available protein sequences. So I won't go through this section today, but as with all the resources, there are other materials available online and on YouTube, which will describe what each of these sections are in a little bit more detail. And UniRef. So UniRef, or the UniProt reference cluster, provides sequence clusters based on sequence similarity. So this is particularly useful if you're doing any kind of evolutionary studies and you want to compare sequences by identity. So as I mentioned, the main resource that I'm going to talk about today is UniProtKB resource. So this exists in two subsections. So we have the Tremble section and the SwissProt section. So all sequence data that we get from sequence repositories goes straight into the Tremble section. And so these entries will be computationally analysed and annotated, but they will not be reviewed by a curator. So, but once a curator looks at these sequences and adds experimental information, these entries are moved into the SwissProt section. So what do I mean by reviewed and unreviewed and annotated and unannotated? So by reviewed, I mean a curator has looked at the sequences, compiled all of the isoforms, if there are isoforms available, for one protein together into one protein entry. They've searched the scientific data, they've searched for publications and added any experimental data which indicates the function of the protein, and they've looked at the sequences to look for any kind of modifications or any kind of annotations that they can add to that. Okay. So in practice, how do we present these resources and how can you access the data? So this is the main UniProt homepage. So at the top of the homepage, you can see there's a free text search bar, which I'll go through in a second. The resources are organised just below this. So clicking on any of these will take you directly to the portal that you're interested in. Below those resources, we have a tools section. So you can do BLAST, you can do multiple alignments. And I won't be talking about this today, but all of the data in the UniProt database is available programmatically. And so there are various videos and tutorials online which will show you how you can access that data programmatically if you wish to do so. So using the free text search bar at the top of the home page allows you to search entire protein entries using either a protein name, or you can search by gene name, disease, or if you know any ontology terms such as gene ontology terms, you can type in the accession number here or the terms directly, and this will bring up a list of all the entries that contain your keyword of interest. So now if you wanted to do a search that was a little bit more specific, so say that you had an organism in mind, you had a protein name in mind, you didn't want every single entry that has your keyword, but you want every entry that has that protein name, you can use the advanced search option. So clicking on the advanced search box provides a drop-down menu with where you can build a more complex search query. So in this example, I've specified a protein name. I've indicated that I'm interested in all proteins with this name that interact with ACE2, and I've begun specifying an organism. So when you specify an organism, there's actually you'll get a drop-down box which will allow you to pick your organism of interest, and then once you're happy with your search query, you select search. And then you'll get a results page that looks like this. So this is a typical results page. So whether or not you search using the advanced search or using just the free text search bar at the top of the page, you'll get a results page that looks a little bit like this. And this is fully customizable. So you can choose what kind of information you want to appear in your results page. So this is just a basic summary. So I've got the entry name, the protein name, the length, the organism, but you can adapt to this. So you can also show function comment, you can show some, say, your location. It's very easy to do that by clicking on the columns. Also, if you haven't applied any filters, so in this example, in the end, I didn't search for a specific organism, so I have the option to do that, looking at the filters on the left-hand side. I can also search for filter via reviewed and unreviewed entries, so I can select only the entries which have been reviewed by a curator, if I wish to do so. So in practice, how does this work? So using this very basic schematic of an autophagy pathway, I've got a list of proteins which are involved in one of the many pathways. And so I can see the search for these gene names individually using the free text search bar, or I can use the advanced search to search by organism, and I can just do one individually. However, if we go back to the main homepage and we click on Retrieve ID Mapping, it's possible to search for all of these proteins in one go, so without doing them one by one. So if you click on the Retrieve ID Mapping tool in the just below the search bar, you'll get a page that looks a little bit like this. So in the box at the top, you can list your gene names or identifiers. And then below that in section 2, you have an option to select your parameters. So in this case, I'm looking for the UniProt entries that correspond to the gene names that I've put in the box. And I've also selected human because I would like to look in human. So this is what the results page looks like. So it's very similar to the results page we've seen before. So I can see here that with those parameters and with those gene names in the box, there are 29 UniProt entries that correspond to those 3 gene names. And if we look at the filters on the side, I can see that 3 of those entries have been reviewed by a curator. So that means a curator has looked at these sequences and added any functional annotation to the entries. And 26 are still tremble entries, which means they're still unreviewed and a curator hasn't looked at them yet, but they still contain computational annotations. So moving on from this, so say I don't know the gene names, but I have a list of identifiers. So these could be UniProt identifiers or accession numbers as we call them, or they could be RefSeq identifiers, PDB identifiers. It's possible for us to search for entries by just using the identifier alone. So again, using the Retrieve ID Mapping tool, we can convert identifiers into gene names. So in the same way that we typed in a list of gene names into the search box, if you type in your list or copy and paste your list of UniProt accession numbers or identifiers, and in the parameters in section 2, if you type in UniProtKB AC to ID to gene name, because we want to go from the UniProt accession number to the gene name, and you press search, you'll get all of the gene names that correspond to your accession numbers. So you can actually use this tool to convert non-UniProt identifiers to UniProt identifiers and vice versa. So say if you've got a list of RefSeq identifiers and you want to know the corresponding UniProt identifiers, you can use this tool to do that conversion either way. Right, so now, okay, to build on that, so say that you don't have the gene name, you don't have the protein name, you don't have any identifiers, but you have a string of amino acids. It's possible, so using the peptide search tool, you can actually find protein entries which correspond to a specific string of amino acids. So again, clicking on the peptide search tool function at the top of the homepage beneath the free text search bar brings you to a page that looks like this. So in the search box, you enter your peptide sequence, and if you wish, you can specify a specific organism. So in this example, I've specified human, and then you click search, run peptide search. So this tool will bring up all of the protein entries that contain this specific string of amino acids. So in this example, I can see that there are 3 protein entries that contain this specific string, I can see that 1 has been curator reviewed and 2 have not. Right, so until now, so I've shown you how to search for proteins in different ways, and I've used this term entry quite a lot, but I haven't actually said what's an entry and what's in an entry. So we use the word entry to describe a protein record, so it's a record of all the information about that protein that we have. And so you can access what's in a record by clicking on the accession number. So in this example, I'm clicking on the accession number of a reviewed entry. So that means a curator has looked at the sequences and they've added any functional information that they've got from publications. So clicking on the accession number of that particular entry takes me to a page that will look like this. So there are various ways in which you can view this entry. So the default is to view the entry as a whole. So you'll get beneath the display options at the top, you'll get a list of quick access tabs which allow you to explore different sections of the entry that you're interested in. You can also display all the publications. So if you click on the publications list below entry, you'll get a list of all the publications that mention your protein of interest, and we indicate how we've used that publication. So we cite where we've taken functional information, subcellular location information. There's also so there'll be a webinar coming up, I think next year, about community curation. So it's actually possible for you to add annotations to your own papers for us to have a look at and include in entries. So and I think there's a blog and there's various online resources that will tell you how you can do that. So that's quite a useful thing to be able to do. And the feature viewer I'll go through a little bit later, as the featured table as well. So looking at the quick access tabs. So if we click on function, for instance, you'll get a brief description of what the physiological role of the protein. If the protein is an enzyme, we'll indicate all the reactions that it catalyzes and indicate the EC number. And you'll notice that at the end of each section, there's little yellow tabs. So these indicate the source of the data. So we cite where we get any functional information from. So all of these tabs are clickable. So clicking on the drop-down menu will give you a list of the publications that we've used to get this data. So then you can go back to the source and read it properly for yourself if you wish to do so. So again, as I've mentioned, please feel free to ask any questions in the chat box. You're very lucky because Rosanna is our enzyme expert and she will be she's available to answer questions today. So please do ask any questions, and anything else that's that comes up, I'll happily answer them and answer the questions later on afterwards. Right. So clicking on subcellular location tab will take you to a subcellular location viewer, which will actually show you where your protein of interest is expressed in the context of the cell. And in this example, so we've got this data not from publication, but we've actually inferred it by orthology, which is the case sometimes, and we actually provide the source of that ortholog as well. So if you click in the drop-down box, there's an accession number there which you can click on. That will take you to the entry which has the primary source of that information. Clicking on the interaction and structure tab will take you to sections which will indicate any interactions that your protein may be involved in. Throughout the whole entry page, we actually have a list, various cross-references that you can click on that will take you to other databases which will provide more specific information on interactions or structures or anything else that you may be interested in. If there's a crystal structure available, we'll provide a list of those structures with direct links to the PDB resource. I'll briefly mention the structure viewer again a little bit later. So I've shown you how to search for a protein and what's in an entry page, but I haven't talked to you about protein sequences, which is the first thing that I began talking about. So every UniProt entry has a protein sequence attached to it, and will indicate any isoforms as well, any variants, polymorphisms, sequence errors, processing information such as cleavage sites, and we also provide various sequence analysis tools as well. So I'll only mention BLAST and ALIGN today, but there's actually going to be a more detailed webinar specifically on BLAST and ALIGN in a couple of weeks' time. So please do listen to that if you want a little bit more information on that. We also have YouTube videos as well that you could watch beforehand. So using spike glycoprotein as an example, if we click on sequence, you'll get the protein sequence. So you get the amino acid sequence, which is interactive. So you can download the sequence in FASTA format. You can add the sequence to a basket. You can actually, from this view, you can actually directly blast search this sequence as well if you wish to do so. Clicking on the feature table will highlight specific amino acid features such as glycosylation sites, any disulfide bonds, any topological information as well, and these features are listed all in one table. So clicking on the feature viewer will actually show you these features in the context of each other. So in this example, I've highlighted a specific region, and I've clicked on a site that has been a site that which has been altered by mutagenesis, so site 501, and I can see that that site actually is within a receptor binding motif. And so that's quite useful to see how the different features correspond to each other and whether or not they fit within specific domains or regions of interest. Below this, if there are any structures available, any sites that you click on or any regions that you click on will actually be highlighted in the crystal structure as well. And this crystal structure is versatile, and it's interactive, so you can move it around to look at specific features or specific areas that may be of interest. And there's a direct link to PDB if you want to see some more information on that. So I've mentioned BLAST very briefly. So BLAST can be accessed via the main homepage, so just below the search bar, or via the protein entry itself. So the BLAST search allows you to find similar proteins based on the sequence. So clicking on BLAST will take you to a page that looks like this. So here you can search for similar protein sequences using this tool. And so in the webinar that's coming up in the next couple of weeks, you'll get a little bit more information on this and on the parameters and how you can make the most of your BLAST search. So I'll just briefly explain that you can do it. If you were to BLAST search your protein of interest, you'll get a page that looks like this. So here we list proteins which are similar according to the protein sequence. Then the identity column, we show you the percentage identity, so this shows you how similar your proteins are from your query sequence. So using the BLAST page itself, you can actually align two or more sequences that are similar. So if you identify two sequences, so if in your BLAST search you identify sequences which seem to be highly similar, you directly from this page you can align them. You can do a multiple alignment. Otherwise, if you click on align next to the BLAST option in the toolbar, it will take you to a page where you can copy and paste your sequences of interest and align them there. So an alignment, so once you do an alignment, it will look a little bit like this. So this aligns your sequences according to identity. So the stars beneath the amino acids indicate sequences which are identical. So you can see whole regions which are identical in this example. We offer the option to click on various sites of interest, so you can look specifically at glycosylation sites, mutagenesis sites if you're interested in doing that. So next, I'll show you very briefly how to access proteomes. So as I mentioned earlier, a proteome is the entire list of proteins expressed by an organism, and it's possible to access these protein lists via the main UniProt homepage by clicking on the Proteomes portal tab. So UniProt has over 160 proteomes representing species belonging to the 4 superkingdoms. And as I've said, a proteome contains a list of proteins thought to be expressed by an organism. And we have proteomes for species with completely sequenced genomes, and we're always updating this. So please do submit any sequences that you have. Each proteome has a unique identifier, which you can search for using the search free text search tab. And as I mentioned, this is always being updated, so please do email us if you have any questions on this. And there are various resources available which will provide a little bit more information on any of these and on anything that I've mentioned so far. Right. So in the last section of this webinar, I'm very briefly going to go through disease and mutagenesis data and how we present this. So disease information can be accessed via the main homepage by clicking on the disease tab. Alternatively, we present human disease information in human entry pages themselves. So clicking on the pathology and biotech section in the left-hand filter will take you to a list of diseases that your protein of interest may be involved in. So now for model organisms such as C. elegans and Drosophila or Xenopus, we don't actually provide disease information, but we do provide a section which we call disruption phenotype, which describes phenotypes of null mutants and phenotypes that arise as a result of single, double, multiple mutations, or as a result of RNAi or morpholino knockdown. And if it's relevant, we'll indicate the developmental stage. We also list mutations that disrupt one or multiple amino acids in the feature table and the feature viewer. So in this instance, we'll provide the amino acid affected. So if it's amino acid that's been substituted for another, we'll indicate that substitution. We'll provide the gene name, the mutant name if known, and we'll indicate if the phenotype is due to a mutation of several amino acids as well. And again, publications are cited at the end of every section, so you can see the source of this data. So that was a very quick summary of the UniProt database. Please do tune into our upcoming webinars for more information. So hopefully you will have seen how to perform a basic search, how to access functional data and sequences. You'll see how to analyze specific sequences and explore proteomes, and how to access disease and mutagenesis data. The other thing I'll mention is that UniProt releases every 8 weeks and is freely available, and data is provided in a range of downloadable formats. Please do email us at help@uniprot.org if you have any specific questions, or if you notice anything that you would like to see, or if you have any queries about anything that you're working on, or any of the data in the database. Please do email us. And I realise I've gone through quite a lot in a short amount of time, so please feel free to watch this video again if you wish to do so. And we provide additional training materials online and the training website and also on YouTube, so please do have a look at those. UniProt funding comes from a range of institutes, so I won't go through all of this now, but have a look if you wish to see. And the UniProt team is quite huge, so there's a lot of people doing a lot of work for this database, and we're only as good as our users make it. So please do interact with us and tell us what you need and what you would like to see from the resource and how you use it, and let us know if we can help you further. And thank you very much for listening.
Thank you very much, Emma, for today's webinar. All right, so now we will go through the questions.
So someone has asked how data representation in UniProt is different from GenPep, which is more advantageous. So I would say it depends on your needs. So I don't work for GenPep, and I'm not really familiar with the different data types that they present. But I think every resource has its own advantage, and it really depends on the nature of what you're working on and what information you need to find. And it's usually the case that you need to use a range of resources to find out what it is that you're interested in. Which is why we provide cross-links throughout our entry pages so that you can access more specific information, so you can actually use a combination of resources to help you. So without knowing what information you want specifically, it's kind of hard to say which is more advantageous. I would say they both have their advantages without knowing very much about GenPep, but and there's a need for both. So it really depends what you're looking for.
There's a question how those figures are annotated in the curation system. Are those annotations are unique or just for example?
So are these the examples that I presented in the webinar? The way that we, so in the curation system, the way that we curate, we have a list of proteins and we search publication databases to find all the information that we can about our protein of interest, and we have certain criteria that we look for. So we look for physiological functions, and we sift through facts basically. So we don't go by opinion. So we have a way to indicate if it's an author opinion, but we look for proven data. So we look for evidence. So we look at the experiments that have been done to prove claims made by researchers. We look at the figures to see whether we trust the data, and everything that we write, some of it's free text, some of it is according to certain specialized vocabulary, but we make sure that we cite all the information that we get, that we read. So every function comment, every sentence in a function comment should have a reference or some indication of where the data comes from. We don't assume knowledge, so we try to indicate where possible where everything comes from.
We have a couple of more questions.
Sure.
One is about the algorithm, what algorithm peptide search uses, and another one is the reviewed portion. So who is reviewing?
Okay. So I think the algorithm used behind the peptide search, I'll have to get back to you about that because I'm not a programmer. But if you have a look online, there will be various resources and various tutorials which will describe to you how it works. Have a look on the main UniProt homepage as well, but I can try and get someone to get back to you about the algorithm if you wish to know some more information on that. And the second question, the reviewed portion was reviewed by whom? So that's reviewed by us, the curators. So anyone who's employed as a scientific database curator will be a reviewer, basically. So that means, so by reviewed, we mean reviewed by us curators. So we've looked at the protein sequence, we've searched for all publications that mention your protein of interest or have any information about the sequence and where it's derived, any kind of modifications. So it's quite a thorough reviewing process that we go through. So it's usually based on lots of sources. It won't just be based on one publication or one review. We look at the primary publications and the primary research. Thank you, everybody.